Dear students, you are welcome back to STR, Shivarajan's teaching room. So there is a request to you. The viewers may please subscribe this channel and press the bell button also. So in the last chapter we have been discussing about flowers, and we had seen that a flower has four fundamental parts: calyx, corolla, androecium and gynoecium. Androecium is the male reproductive part and gynoecium is the female reproductive part. Okay, today in this episode we are going to discuss about the male reproductive part, that is the androecium. And the androecium is a group of stamens. The androecium consists of a group of stamens. And a stamen has two parts: a long and slender stalk, a long and slender stalk called the filament, and a box-like structure at its tip called the anther. So a stamen has two parts: a long and slender stalk, it is called filament, and at the tip of the filament there is a box-like structure, what is called as anther. And if you observe this anther, we can see that it has two lobes. It has two lobes like this. This is one lobe and this is a second lobe. And both the lobes are connected by a connective tissue in the center. So there are two lobes in the anther. The anther has two lobes. So this is one lobe, this is one lobe, connected by the connective tissue in the middle. So the anther can be called as bilobed. The anther is called bilobed because it has got two lobes. Bi means two, so it has two lobes, so it is bilobed. And if you observe a lobe, we can see that it has two theca. It has two theca. Each lobe has two theca. This is one theca, this is another theca. So if we cut the anther in the cross section, see, this is one lobe, this is one theca, this is another theca. This is another lobe. It is like this. This is the connective tissue. So this is a lobe and this is a lobe. Each lobe has got two theca. Theca means cavities or spaces inside it. So we can say that it's bilobed and dithecous. Theca, T-H-E-C-A, theca means cavities or capsules or compartments, we can say. So it has got two lobes, so it is bilobed, and each lobe has got two theca, so it is called a dithecous. So an anther is bilobed and dithecous. And the anther has got four cavities. An anther has got four cavities running lengthwise from here to here, running lengthwise. There are four cavities at the four corners. At the four corners of the anther, there are four cavities running lengthwise, and these cavities are called microsporangia. Microsporangia. So what are microsporangia? Microsporangia are small cavities running lengthwise in the four corners of the anther, and they are the microsporangia. And now we will see the structure of a microsporangium. We will see the structure of a microsporangium. See, this is the structure of a microsporangium. The structure of a microsporangium. It has got 4 microsporangia at the 4 corners. So each microsporangium has got 4 wall layers. Each microsporangium has got 4 wall layers. This is the outermost layer. It is the epidermis. So this is the epidermis. Like that, the complete thing. So the second layer is called endothecium. The second layer is endothecium. And the third layer is having more than 1 layer of cells. It is like this. So this is the third layer. It is made up of more than 1 layer of cells. It is called a middle layer. Middle layer. And the innermost layer, this is the innermost layer. It is called tapetum. So what is the structure of microsporangium? This microsporangium has got 4 wall layers. The outermost layer is the epidermis. So this is the epidermis. And the second layer is called endothecium. Epidermis and endothecium are made up of 1 layer of cells. But the third layer, which is called the middle layer, is made up of 2 or more layers of cells. So the outer 3 layers are epidermis, endothecium and middle layer. And the innermost layer or the 4th layer is the tapetum. The tapetum is also made up of 1 layer of cells. In the tapetum, the cells are filled with cytoplasm or rich in cytoplasm and also rich in stored food, and each cell is having more than 1 nucleus, that is multinucleate. So tapetum, what are the special features of the tapetum? Tapetum is made up of 1 layer of cells. The cells are filled with the cytoplasm, rich in food stored or rich in food materials, and it is multinucleated. And the function is inside this cavity, it is inside this cavity that the pollen grains develop. And the developing pollen grains get nourishment from the cells of the tapetum. So we once again go into the structure. The microsporangium has got 4 layers, 4 wall layers. The outermost layer is the epidermis. The second layer is the endothecium. Epidermis and endothecium are made up of a single layer of cells. And the third layer is the middle layer. It has got 2 or more layers of cells. And the innermost layer is the tapetum. The tapetum, the cells of the tapetum are rich in stored food. It is filled with cytoplasm and they are multinucleated. These are the special features of the cells of the tapetum. And the function of tapetum is it gives or provides nourishment to the developing pollen grains inside the microsporangium. So this cavity inside, the very central cavity, it is the microsporangium. It is inside the microsporangium that the microspores or the pollen grains develop. The developing microspores or the developing pollen grains get nourishment from the cells in the tapetum. So to provide nourishment to the developing microspores is the function of tapetum. And what is the function of epidermis, endodermis and middle layers? These outer 3 layers are the protective layers. The outer 3 layers are the protective layers. They protect the microsporangium and also helps in dehiscing. Dehisce means to break open. Dehisce means to break open. So the epidermis, endothecium and middle layers are the protective layers of microsporangium and they help in dehiscing the anther to disperse the pollen grains. So when the pollen grains are matured, the microsporangium breaks The breaking is called dehiscence, and dehiscence of the microsporangium is done by epidermis, endothelium and middle layers. So what is the function of these outer three layers? These outer three layers, epidermis, endothelium and middle layer, are the protective layers of the microsporangium, and they also help in dehiscing the microsporangium to release or disperse the microspores or pollen grains. So, and the tapetum is the innermost layer. It is made up of one layer of cells. The cells are rich in food, stored or rich in food materials, and the cells are multinucleated. They provide nourishment to the developing pollen grains inside the microsporangium. So we have seen the stamen and microsporangium. Okay, a stamen is bilayer. A stamen has got two parts: a long, slender filament and a box-like structure is tip called the anther. The anther is bilobed and it is dithecous. And at each four corners, there is a space or capsule-like structure which is called as the pollen sac, or it later develops into the microsporangium. And this is the structure of the microsporangium. So we have seen the stamen and the microsporangium. So in the next section, we'll be looking into or we'll be discussing about the microsporogenesis. What is microsporogenesis? The formation of microspores from the pollen mother cells inside the microsporangium. The development of or formation of microspores or pollen grains from the pollen mother cells in the microsporangium after meiosis is called microsporogenesis, and that we will discuss in the next episode. Okay, thank you.